all these cells in our body need to be able to replicate themselves. They need to be able to split up. And they do this through a process of DNA replication. For humans, the semi-conservative process, because it's half old and half new DNA. So there's the old DNA that gets split up, and then there's new DNA that gets added to it and completes the DNA. So we have two separate strands and allows our cells to be split up into two separate pieces. And they do this all along the DNA in multiple areas at once in these replication bubbles. So at each of these replication bubbles, there's this area called a replication fork, and that's where all of the work is being done. Each of these little replication forks is getting busy, doing all kinds of work. So what's going on is here we have our fork, and we have our nitrogenous bases, A, T, T, A. And since our DNA runs in a double helix pattern, we have to be able to split that up. And we do that through a protein called helicase. Helicase separates our DNA and unwinds it and gives us these pieces to work with that we then have to add to. Let's just say that this is 3 prime and this is 5 prime, so that means this is 5 prime and this over here is 3 prime. Now there's this other protein called DNA polymerase. Now what it does is it follows along in a 5 prime to 3 prime direction, okay? And it basically finds all the matches to the DNA. So pretty easy for this leading strand, we call it, because it's nice and smooth, leading strand, so that this is 5 prime and this is 3 prime. However, over here we call this the lagging strand. Guess I'm hungry. We call this the lagging strand because the DNA polymerase has to run in this direction here, okay? But it can't run as smoothly as it does here, because as soon as it follows that pattern, right, follows along the DNA and finds all the matches, this helicase has already split up more DNA. So what it does is it has to come back over and over again on itself. Now, these different fragments are called Okazaki fragments, and each of these fragments are then held together by this extra protein called ligase. Now what ligase does, it kind of acts as a glue. So it smooths out those edges, it sticks those pieces together, and makes it so that the DNA here is still the same as over here. It's just having to do a little bit more work. And that's DNA replication.